for the first practical. Today we're going to do connective tissue. This is kind of going to piggyback. That's all right. We're going to kind of piggyback off of what we did last week with the epithelial tissue and get everything wrapped up because next week practical one. So, it's, I told you it would get here quickly, and it has. So next Thursday at midnight, or Wednesday night, Thursday morning, however you want to look at it, your first practicals will open up. You'll have until 11:59 on Thursday evening to sign in at some point, and either knock them both out back to back, or if you want to do one in the morning, one in the afternoon, both of them in the evening, whenever you want to do them. It's just it's up to you to get on sometime next Thursday and take care of the 50 question anatomy test and the 25 question physiology test. Now, when it closes Thursday night, normally I would work on the grades and work on the anatomy tests on Friday. Unfortunately, with the holiday coming up, I do have plans to go to Georgia to go visit my folks, and so we are leaving at like 5 o'clock Friday morning. So needless to say, I will not be available to grade the practicals on Friday while I'm driving 7.5 hours to Georgia. Nor will I be able to grade them on Monday when we come back, or the time in between because we'll be visiting. So I will get your exams graded on Mardi Gras Day. We'll be back on Monday. There's nothing going on for Mardi Gras really, so there's no reason for me to be doing anything else. So I'll probably spend Mardi Gras Day just grading exams. I got 3 classes on Thursday. Everybody's got practicals, so I'll get everything graded on that Tuesday after. So the reason I'm telling you that is because normally I'll get your exams graded the next day, if it's a Thursday exam, because I have office hours usually on Friday morning. But unfortunately, I've had this trip planned for a while. So I'll get them done as soon as I get back. But I just wanted to let you know that way you weren't constantly checking Canvas all weekend, to see if they were graded and emailing me, Dr. B, when are you going to have them graded, all that kind of stuff. So you can take it on Thursday, be done with it, relax a little bit for the weekend, and I'll have them back to you as soon as I get back in town. The week after that, we will not meet. The week after that is the Thursday after Mardi Gras. Mardi Gras holiday for everybody is Saturday, next Saturday, the 13th, through Wednesday. So because the Monday, Tuesday, Wednesday labs don't meet, and we try to keep everybody on the same page, typically that Thursday would be utilized as kind of a study lab if we were going to meet in person, where we might get together and do, some type of, physiology activity or something like that. But since we're doing this virtually, I'm just going to give y'all, the week off and let you kind of mentally recover from the practical the week before. So after today, I really won't see y'all until the 25th, because I won't see you next week because you'll be taking your practical, and I won't see you the week after that because it is Mardi Gras. So there's a little bit of a gap here that I want you to take advantage of to kind of mentally recover before we dive really into this head first and very rapidly. But I do want to point out, don't don't let the due dates kind of slip past you. When there's a lull in the routine here, oftentimes students will say, oh well, I've got plenty of time to take care of my pre-lab for exercise 4, and the homework assignment, which is bonus, and the physiology assignment, which is bonus. So in between taking the practical next week and then the next time we'll meet, you have 2 bonus assignments and the pre-lab for exercise 4. So definitely don't just forget about that stuff. Knock that stuff out next week, maybe when we're not, or the week after next when we're not meeting for during Mardi Gras week or something like that. But don't wait till the last minute, when you're trying to scramble around, or you just forget about them, completely. Because those extra credit points, are something to look forward to, and are worth significant percentage on top of your score. The homework assignment is going to get added onto your anatomy score, so it's worth up to 6 points. And the physiology that I showed you before about the from the first lab, that's worth 6 points on your physiology test. So there's 12 extra credit points sitting out there. So make sure you take care of those assignments. I don't want anybody to miss out on that. Any general questions before we get rolling? I'm going to walk through Canvas and kind of show you all a couple of things as to what I'm going to be using today as we go through the lab, kind of like what I did last week. But is there anything in general anybody has questions about?
Where is the bonus exactly on the in Canvas?
It's in Canvas. I'll show you. I'll show you in just a second here. Um, Anything else before we jump over to Canvas? All right.
No.
Well, let me pull up Canvas here, and I'll show you where you can find everything we're going to use today, and the bonus work and all that kind of good stuff. The first was the physiology bonus, and that was this physiology experiment on homeostasis that I kind of pulled up, and we talked about it. It entails you taking your heart rate, your respiratory rate, doing a little minute worth of exercise, and taking it again, and drawing a chart and a graph. This is all about completion. All right, so you're going to do this experiment. You're going to submit the lab report to me. The directions are in the activity. And if you do it and it looks good to me, I'm not going to grade it necessarily for correctness because it's your own data. But if you do it and you get it to me and then you've done it correctly, then you're going to get 6 points on the physiology test. The other one is homework one. Homework one is down here, and it begins what we're going to be covering for test two. And this is basically kind of getting us ready, or getting you ready for what's coming up in exercise 4 and exercise 5. So you're going to do this bonus quiz right here on homework one. You'll find homework one in the lab manual. All right, it's going to be right after the connective tissues. So you'll use that homework section to answer the questions on this quiz. If you get a 6 out of 6, then you get 6 points on your anatomy test. This one is actually graded. If you get a 4 out of 6, you get 4 points, and so forth. So, you want to make sure you do this quiz by the 24th. The physiology I have set to be turned in by the 18th. So you want to make sure you take care of that first, since that's been open since the beginning of the semester. And then you'll also have your post-lab exercise quiz due next Wednesday before the practical. And then you'll have your pre-lab due for exercise 4 also on the 24th. So on the 18th, you have your physiology extra credit. On the 24th, you have your anatomy extra credit. And on the 24th, you have your pre-lab. Any questions about what you have kind of going on the next couple of weeks and everything? To find the exams, you can either come up here, all right, to the exam module here and click on test 1. That's the anatomy test. Click on physiology 1. That's the physiology test. Or you can do it from the home page. Or probably next Thursday, I will get on, post an announcement Thursday morning, says, hey, your exams are available, and here are the links to them. Good luck, that kind of stuff. So, you'll be able to find them multiple places, and all you have to do is log in through Respondus and get going with it. Now, remember, once you begin the exam, the time's going to start, right? So you have 60 minutes for the anatomy test. And then when you start the physiology test, you'll have 35 minutes for that 25 question test. And it begins the second you start the first question. So, I've stressed this before. I can't stress it enough, especially when you take this anatomy test. make sure your internet is perfect. Make sure your connection is the best it can be, whether it means you got to sit right next to the wireless router, or whether you got to hardwire into the wireless router, or whether you got to make sure that everybody is off any type of streaming or PlayStation or whatever. Make sure that all your bandwidth is being used for your computer, because this anatomy test is pictures, all right? And so it takes a little bit of time for these pictures to download. So you want to make sure that nobody is streaming and nobody's playing games and this and that, and your internet is running slow, or your Wi-Fi is running slow in your house because people are doing that. Now the physiology test, there's not going to be pictures associated with it, so it'll load more quickly. But people, students tend to have problems with this, and if you can't load it and the pictures don't come up, you're going to have to log out and sign back in and try to reload it, and that's just eating up valuable time that you could be using on the practicals. So make sure that you're in a situation where your connection is going to be as strong and as stable as possible. All right. Any questions about the practical, all that kind of good stuff, where you can find it, all of it? All right.
The regular test, the other test is going to be multiple choice.
The physiology test is 25 questions, multiple choice. The anatomy test is the 50 questions, fill in the blank.
Okay, I got you. Thank you.
All right, sure. And like I said, normally the physiology test, I'd have those grades up almost immediately, because there's nothing I have to hand grade. But the anatomy tests take me a little time because I have to hand grade them. So that's why I said don't stress over them, and just wait. I'll have them up on the following Tuesday when I get back in town, and you'll be able to see. You'll be able to see what you got. Now, these will grade themselves. When you finish the physiology test, it's going to give you a score out of 25. So, it'll tell you 22 out of 25 or 20 out of 25 or 10 out of 25 or whatever your grade is, it'll tell you. So you can calculate, multiply that by 4 and you'll know what your raw score is. But the anatomy test, it's going to give you a score, but that's marking everything wrong. That's marking, stuff wrong for misspellings. That's marking things wrong if you capitalize something that wasn't capitalized in the key that may be completely correct. So, whatever score it says on your anatomy test, expect that it's going to, increase, because as I go through and hand grade, if you misspelled something and marked it completely wrong, you lost 2 points. Well, if you got it right and it's just misspelled, I'm going to give you 1.5 points of that credit back. You're only going to lose 0.5 point for not getting it right. So, keep that in mind when when the score pops up at the end of this test, that it's going to, it's going to increase as I go through and hand grade them, unless you just left everything blank. Obviously, if it's blank, then it's wrong, because, I can't give you partial credit for something that isn't answered. But I'll go through and hand grade those as soon as I get back in town.
So we would need to study the diagrams that we have for chapter 1.
Everything we've done so far, all those the models in chapter 1. When in chapter 1 I told you to use this model packet right here to practice on, the muscle man models and this and that, and you could identify the body regions on here and directional terms and all that kind of stuff. Yeah, absolutely. Don't forget about all this stuff, because all that's on it, as well as what we did last week with epithelial tissues and what we're doing today with connective tissues. Anything else before we get going? No, just how to remember it all, right? All right, so today I am going to be using a PowerPoint like I did last week. The PowerPoint I'm using is this one right here, this tissue walkthrough of connective. Just like last week, we had one for epithelial tissue. I'm going to use this one today to show you examples of the different types of connective tissue. The tissue repository, which is right here, I'm also going to pull that up later on, and I'm going to go through when we're done and show you some different examples of the connective tissues that we're going to talk about today, you know, that you could maybe, you know, you should maybe go through and take a look at as well. It's the same tissue repository we used for last week. It's not
Uh,
one specific for this. It's a mixture of epithelial and connective and all that kind of stuff.
Mr. B,
Yeah.
sorry to interrupt, but just a quick question. So when you post, you told us, I think I'm trying to recall, you said if with the tissues, you're not gonna put like a tissue where it's literally almost impossible to identify it. You're gonna put like
No,
tissues that are,
no,
if we understand, if we
it'll be good slides.
it's
Like
like,
this adipose tissue slide right here.
mm-hmm,
It's not the greatest because it's kind of faded and you can't really see. This dense regular connective tissue slide, you know, this isn't a really good example because you know you really can't see the tissue. So no, the tissues that are on the exam would be, you know, something like this elastic cartilage right here. You know, that's a great elastic cartilage slide. So, you know, there'll be very good examples of the slides, you know, and there'll be probably some examples maybe that come from here, some of them that maybe came from the PowerPoint, but they're not going to be pictures that we go grab off of Google or, you know, anything like that. You know, it'll be stuff, you know, from the resources that we have available to us that we make available to you. Now, I mean, I can't just go through and go, okay, well, this is going to be the picture of simple squamous, and this is going to be the picture of stratified squamous, and this is going to be the, I can't, you know, it's not just a memorization thing. You know, you have to be able to recognize the tissue. But yes, the quality of the tissues and the quality of the pictures will all be something that, you know, you won't look at it and go, I can't tell what this is because this is a terrible image. No, they'll all be good pictures. So that answers your question?
yeah, thank you so much.
Sure. All right. So with that said, let's dive into our connective tissue. So everybody knows where to find everything that you're going to utilize in here. And look, also, like I said, I would go back and utilize some of your pre-lab exercises. You know, if you've done the exercise and you know your grade is 10 out of 10, go back and do the pre-lab stuff. I mean, you have unlimited tries. It's going to keep the highest score. So if you've already gotten 10 out of 10, you could get a 5 out of 10. It's not going to change your score. So, you know, you can utilize that too as a study tool if you want. All right, let's get back to the beginning of this and let's talk about connective tissues. Last week when we talked about epithelial tissues, we said had a set of unique characteristics to them that, you know, really defined epithelial tissue and gave you something to kind of go by when you were identifying epithelial tissue. You know, I said, hey, if you look at a slide and you can't pick out what it is right off the bat, then, you know, you can look for these things and it'll at least narrow it down to some epithelial tissue. Well, the same thing is going to kind of apply to connective tissue. And in some cases, it's going to apply to connective tissue as a whole. And in other cases, it's going to apply to certain types of connective tissue. Connective tissue is pretty diverse. Its diversity comes from you know, the way that it's built, and then how it's built is going to give us, you know, an extreme amount of diversity when it comes to the type of functions that we see associated with connective tissues. And that's going to come from the number of cells that we have, or the types of cells, types of fibers, and the type of ground substance that we find in connective tissue. Connective tissue is going to differ from epithelial tissue in a few basic ways. One, with epithelial tissue, we saw cells that were packed very tightly close together. There was little to no space between those cells. And with connective tissue, we're going to see the opposite. All right, we're going to see cells that are sporadically placed, like one here and one here. So there's distance between those cells. And, you know, we call the space between the cells the extracellular space. That extracellular space is not just going to be empty, right? It's not, when we talk about space, it doesn't mean that there's nothing there. It just means there's a distance between the cells. So, you know, that's a pretty obvious difference between the two different types of tissue that we've talked about. The other thing is that with connective or with epithelial tissue, we said that they were all going to have a free or an apical surface. Connective tissue isn't going to have that. And part of the reason why, connective tissue isn't going to have that is because epithelial tissue makes all of our free and apical surfaces. They make all of our coverings. They make all of our linings. Connective tissue is going to kind of be the behind the scenes, because it's going to be underneath the exterior surface. We don't have any connective tissue that's touching the outside world. Everything on the outside world is going to be epithelium, and then underneath that is where we're going to find our connective tissue. Now, connective tissue is going to, for the most part, be pretty well vascularized. We do have some instances where we have tissue that is not very vascular, like with our dense regular connective tissue, which would be our tendons and ligaments, and with our cartilage. But for the most part, connective tissue is going to be pretty vascular, which makes sense because, we know that connective tissue is going to be what we use to attach epithelial tissue to. So anything we're attaching epithelial tissue to, we know has to be pretty vascularized because that epithelial tissue is avascular. But, like I said, we will have instances where we're going to see it be in an avascular kind of setting. But we'll explain how, those types of tissues do get their nutrients. And we'll talk about what that vascularity or lack of vascularity means. We mentioned that connective tissues were going to be made up of three basic components. First of all, we have cells, all right? And the cells are going to be sometimes very specific to that type of connective tissue. And then other times they're going to be cells that we find in lots of different types of connective tissues. The rest of what we're going to have is going to be called the extracellular matrix. And the extracellular matrix is going to be made out of different types of fibers. These are going to be protein fibers and what we call a ground substance. So we're going to have these sporadically placed cells. Between the cells, we're going to have fibers that are going to run in certain patterns, in certain arrangements with different types of fibers. And then we're going to have what we call the ground substance. And that's going to kind of fill in the space around the cells and around the fibers. So, we have a few different factors here which are going to contribute to different configurations and different presentations that we see of these particular types of connective tissue. Depending on which fibers we have, how many of each fiber type we have, how the fiber types are arranged, and then depending on what the ground substance is around it, is going to give us a lot of variability that we can see with this connective tissue, and that's what helps to create the diversity with connective tissue. Now, we mentioned cells, and we mentioned that there were different types of cells. Some of our cells are going to be specific cells of just a single type of tissue, meaning that they only exist in that tissue type. For example, anything we see with the beginning of the word, the prefix of the word being osteo, is going to be related to bone. All right. We don't find any osteotype cells in cartilage. We don't find osteotype cells in the skin. Okay. Osteocyte type cells are going to be limited specifically to bone. So those would be fixed cells. Chondro type cells. Okay. Chondro is referring to cartilaginous cells. They're only going to be found in different types of cartilage. We won't find them in bone. We won't find them in the kidneys. We won't find them in the liver. We're not going to find them in the bloodstream. Okay. Chondro means cartilage. So these would be examples of fixed cells, meaning that we won't find them anywhere else. Now, with fixed cells, we can determine the function of fixed cells based on the suffix of the word. All right. Prefix is at the beginning, suffix is at the end. So if the suffix is blast, then these cells are considered to be building cells. Now, we can have blast building cells, okay, that aren't necessarily a fixed cell, but when we see the word blast at the end of a cell, we know that that means that it's a building cell. It's undergoing mitotic division, and it's helping to secrete the extracellular matrix. A clast cell is a cell that is going to break things down. It's going to remodel. It's going to recycle the extracellular matrix around us. So it's going to make the tissue less than what it was. And then we have site cells. And our site cells are our maintenance cells. These are the cells that are not building new matrix, and they're not tearing the matrix down. They're basically helping to sustain the extracellular matrix. Now, for the most part, a large portion of our tissues are in a constant state of flux. They're always building, they're always tearing things down, and sometimes they're doing it simultaneously to maintain what we have. But there's always a group of cells associated with a particular type of connective tissue that's there to sustain the tissue itself, and that's going to be our site cells. So we gave a few examples here of each. An osteoblast, that would be a bone building cell. A chondroblast would be a cartilage building cell. Now we do have a transient cell listed in here too, a fibroblast. Fibroblast is considered transient because we find it all over the place, but it builds fibers. An osteoclast is going to be a cell that tears bone down or remodels bone or resorbs bone. And then we have some examples of some sites. Osteocytes, chondrocytes, these are going to be maintenance cells that are going to help to keep the extracellular matrix at the point at which it is at a certain given time. Now, we talked about fixed cells, transient cells. Like I said, these are cells that can be found in multiple types of connective tissue, meaning we can find them kind of all over the place. And the first one we have listed there is one we just mentioned, and that's a fibroblast. Now, this is a fiber building cell, and we said that all connective tissues were going to have fibers in the extracellular matrix. So we could expect to see some fibroblasts present in the extracellular matrix, because that's going to be what's helping us to secrete these protein fibers. Macrophages. Macrophages are found all over the body, and these are basically phagocytic cells. These cells are designed to eat things, whether it be to eat other cells like bacteria, or to just be to clean up debris. Perhaps we have some damaged or destroyed tissue, and we need to clean that tissue up. Then we would have macrophages that were available to be able to do that. Plasma cells. Plasma cells are a type of cell which is going to secrete antibodies. Antibodies obviously are used in immune response because they're going to help to attack foreign pathogens that maybe enter into the body. Mast cells. Mast cells are typically associated with blood vessels or near blood vessels because they're going to secrete a substance called histamine. Histamine is going to cause vasodilation during tissue damage or an allergic reaction, which is going to cause inflammation of the tissue. Adipocytes, these are adipose or fat cells, and adipocytes are going to be found all over the body, and they serve actually a variety of functions to us. Common places we see them are in the skin, below the skin in the hypodermis, but we also find them around internal body organs. We're going to find adipocytes and fat cells in the center of our medullary cavities and long bones. And the purpose of them is to store triglycerides. Now, what we use those triglycerides for and how we use those adipocytes, that's where we're going to start to see a little bit of variation and diversity. And then leukocytes, which are our white blood cells. And leukocytes are also involved in immune response. And depending on the type of leukocyte, which we have 5 different ones, is going to then determine what pathogen those leukocytes are helping to fight off. When you get into AMP2, we'll talk a lot more about leukocytes. And then we have the cells like thrombocytes and erythrocytes, which are other blood cells. But these are the transient cells that I want you to remember for, say, like the physiology test. Now we're going to identify leukocytes and adipocytes, and mast cells are a little harder to see, but we'll take a look at a couple of them. But we'll look at fibroblasts and macrophages and chondrocytes and osteocytes. We're going to look at a bunch of these different cell types today.
Dr. B.
Yep.
If we identify a macrophage as a leukocyte, would that technically not be wrong though?
Well, the leukocytes that we're going to identify, we're only going to identify them in the blood. The macrophage, I mean, yeah, I see where you're going with that. I mean, because, some of the leukocytes are macrophages, but the macrophages that we're talking about, I'm going to identify in like areolar connective tissue. But on the blood slide, just identify it as a leukocyte.
Sounds good.
All right. So connective tissue fibers. We said before that the fibers that were going to be secreted in the extracellular matrix were going to be protein fibers. Now, there's a reason that we utilize protein when we're building things in the body. The reason we use proteins is because proteins are fairly stable molecules. All right, they're pretty non-reactive. They're not going to change their structure. If we don't change the structure, then we're not going to change the function of them. So they're reliable building blocks that we can use to build things like connective tissues and muscle tissues and things like that. Now, proteins, though, on a whole, as a molecule, they are susceptible to damage by one thing, and that one thing is energy. All right. So whether we're talking about heat energy or we're talking about radiation energy, electrical energy, chemical energy, any form of energy can make a change and to the shape and the function of a particular protein, and we call that process denaturing. All right. So when a protein is denatured, it's changed, and that's bad. Okay, that's bad for us. We don't want to denature proteins because we want a protein to work the way that it's intended to work. Now, we've all seen denaturing of proteins done. If you've ever cooked anything, basically in your entire life, the heat energy. Hold on a second. Excuse me. The heat energy. Thank you. That we're applying to the food is serving the purpose to denature the proteins in the food. This changes the physical composition of the food. Whether you're cooking meat or whether you're cooking eggs or whether you're cooking vegetables or whatever, the heat is there to help to break down some of the bonds and like I said, to change the structure of some of these proteins. When we cook meat, the main reason we're cooking meat is to break down some of the protein fibers that we find in meat, which is basically all meat is. From the connective tissue that's in it to the muscle tissue, it's all just protein. The heat energy changes it structurally. An egg, though, is the best example because we can physically see the denaturing take place. If you crack an egg open, what have you got? You got some clear, liquidy, kind of gelatinous, and then you have the liquid yolk in the middle. What happens though when you put it onto a hot pan? What happens to the clear liquid almost immediately when you put it onto the hot pan? It turns solid and it turns white. I mean, it completely changes, and that's what happens when we introduce heat energy to a protein. It changes the structural integrity of that protein, which is going to change the protein itself. And that's what we visibly see take place. As we add more heat energy, we denature more of the proteins. It continues to cook until we end up with all the whites cooked through. The yolk is the same way. It doesn't change the opacity or the opacity. Is that the word I'm looking for? Yeah, of the yolk, but it solidifies it. That's denaturing at its perfect example. Why is energy so harmful to us in all forms? Okay, I mean, of the three types of energy that I just listed, or four types of energy, all four of them are bad for us. Okay, because all four of them can denature proteins, and we don't want that to happen because that destroys tissue. Now, the three types of connective tissue protein fibers we're going to see are going to be collagen fibers, elastic fibers, and reticular fibers. Now, collagen fibers are the most abundant type of connective tissue fiber that we find in the body. Collagen fibers are going to be found in almost every single type of connective tissue. In fact, all three of these fiber types are at minimum mostly found in almost all types of connective tissue, but collagen is definitely the most prevalent. Collagen is going to be designed in a way that kind of helps it to bind things together. Collagen fibers are very strong and very flexible fibers. These are going to be pretty thin fibers, but what we're going to do is we're going to weave these fibers together and we're going to create thicker parallel bundles. So the way I kind of picture collagen fibers is like tiny strands of thread, right? We can take these tiny little strands of thread and we can weave these strands of thread together and we can make a rope, right? The more strands we have, the thicker the rope. The thicker the rope, the stronger the rope, right? So that's kind of what I picture in my mind when I've always thought about collagen fibers, is that there are these tiny threads that get woven into ropes. And we know that ropes are flexible. We know that ropes can be used to bind things together. We know that rope can be used to prevent things from pulling apart. But one thing that rope isn't is it's not sturdy, right? We can't stand a rope up on the end and let go of it and it maintain its structure. It's going to fall down into, you know, a heap on the ground. So we're not going to use collagen fibers necessarily to directly build anything, although we do use collagen in bone to build bone, but we'll touch on that when we get to the skeletal system. But for the most part, collagen is going to be used to give resistance within connective tissue. Elastic fibers. Elastic fibers are going to have elastic properties, and elastic properties are kind of two-part. One is the elasticity, which is the ability to return to its normal shape after it's been stretched, and the stretching part is the extensibility. So we have fibers that can become longer, but then when we let go of them, they will go back to their original shape and size, right? Which is kind of the definition of elastic. I mean, anything that's elastic, that's working the way that it should do those things. I mean, the elastic in your socks, elastic in your waistband, right? What happens if it stretches and it doesn't go back to its original size? Well, then we throw it away, all right? Because it's not doing its job, because its job is to shrink back down to the size that it was for whatever purpose it's trying to serve. And that's what we need these elastic fibers to do. Now, we're going to find elastic fibers in a lot of different types of connective tissue. Sometimes we'll find them in small amounts, and other times we'll see them in much more significant amounts. And obviously, the amount of elastic fibers that we have is going to change the consistency of the connective tissue and give it more elastic properties. The last type of fibers are reticular fibers. And reticular fibers are a specialized type of collagen fiber, but they're not going to be arranged in the same way as our traditional collagen fibers. Instead of having these very thin bundle-like protein fibers, we're going to have very thin, but they're going to be branched, almost like a tree trunk with branches coming off of it, right? We're going to use reticular fibers in the way that we would use 2x4s, all right? 2x4s we use to build things. 2x4s we use to create framework. When you drive by a house that's under construction, you know, the first thing they're putting up is wood, is these 2x4s, all this lumber. Why? Because that's how they determine the size and the shape of the rooms and the structure of the house, right? That's what's at the core of the house. Well, this is going to be the type of fiber that we find at the core of a lot of our organs. A lot of our organs that are structurally designed to be a certain shape and a certain size because we need them to work a certain way. So organs like kidneys and lymph nodes and the spleen and to some extent the liver are going to have reticular fibers because we need these organs to be a particular shape and size and structure in order for them to work properly. So it's the reticular fibers are going to create a framework, so to speak, for those organs, much in the same way that those 2x4s create a framework in your house. Now, again, these three fibers can be found in just about every type of connective tissue, and at least in a little bit. Some of them may not be found in all types, but the way that we mix these together is what's going to contribute to the diversity of the connective tissues. Think of the three fibers like three different spices when you cook, right? If you have salt, pepper, and garlic powder, you have three very distinct spices, three very distinct flavors that if you mix them in certain combinations, they will taste almost completely different, right? And if you add them at different times, they may taste completely different. So it's not only in the amounts that we're putting in, but it's in the way that we're putting them in the food that we cook. The same thing kind of applies to these fibers, right? How many we have, the ratio that we have of them compared to each other, and then how they're put into the tissue is going to contribute to how that specific type of connective tissue works. Which fiber is the strongest? It depends on what you're asking as far as strength goes. If you're asking for strength for separation, then it would be collagen fiber. If you're asking for strength for support, then it would be reticular fiber, right? Kind of in the same way that, it's like I said, it's like comparing a rope to a 2x4. They have two completely different jobs. One is stiff and you can build on, okay? And the other is something strong that you can resist in pulling. Does that make sense? I know that doesn't specifically answer your question, but there's really no way of saying, oh, this one is stronger than that. I would probably, if you had to pick, I would say collagen, just because the way that we can arrange them, right? Because we can have, more and more collagen woven together. I mean, look, you can take threads, right? And you can make a rope. You can make a piece of rope this thick, or you can make a piece of rope, this thick, right? And the more collagen fibers, the more threads you have, the thicker the rope, the stronger it becomes. Dental floss is a tiny little piece of rope, but if you weave enough of it together, you can make something that you could use on a boat. All right. So if I had to pick one, I'd say collagen, but that's just because there's so much more versatility to it because we can continue to add fibers. Reticular fibers are not going to end up, they're not something we're going to build and make thicker. Does that answer your question? Sort of. All right. All right. So what else are we going to see that contributes to the functionality of our connective tissue? Well, that's going to be the ground substance. Again, this is going to be the matrix that's secreted around the cells, which is going to help to bind the cells together. It's going to help to support the fibers and keep the fibers arranged in a certain pattern. So we're going to see a lot of different types of ground substance, and this is going to contribute to a lot of the functionality we see with these connective tissues, because the ground substance can vary greatly. It can be anything in the spectrum almost between a liquid and a solid. We can have semi-solids, we can have gelatinous, we can have complete liquid. We can see some things that are almost plasticky. So it just depends on the combination of the fibers that we have, along with whatever type of ground substance that we're going to be surrounding those fibers with. Hyaluronic acid, this is a thick substance that we're going to find, which is going to help to lubricate our joints. We also find it on the inside of the eye, which is going to help it to maintain the shape of the eye. Chondroitin sulfate, something else that we hear mentioned oftentimes with joint health, because it's going to be one of the main components of our cartilage matrix. Dermatin sulfate, this is going to be found in the skin and the blood vessels and the hearts and the heart valves and the tendons. Keratin sulfate, keratin is going to be one of the things we talk about when we get into the integumentary system. This is a type of ground substance, though, that we're going to find in bone and cartilage and the cornea of the eye. The cornea of the eye is epithelial tissue. Plasma, this is our liquid extracellular matrix that we'll have, and this is going to be what we find with blood. It's primarily going to be made out of water, and we know that we can dissolve things in water, which is going to allow us to help to transport things throughout the body. And then hydroxyapatite, which is going to be a substance that's going to be found in bone that is going to help with the mineralization process to give us our solid extracellular matrix that we find in bone. From an embryonic standpoint, just remember that, mesenchyme is going to give rise to all of our other connective tissues outside of what we find in the Wharton's jelly and the umbilical cord. We talked about the mesoderm, and in fact, in one of the first paragraphs in this exercise, it reminds you that the mesoderm gives rise to all of our connective tissues. So, on the practical, if I say, hey, identify this tissue, and it says, and what dermal layer does this come from? If it's connective tissue, you've only got one choice, okay, and that's going to be mesoderm. The embryonic tissues are going to begin to differentiate from this mesenchymal tissue, and they're going to give rise to the connective tissues that we see as mature connective tissues. Mature connective tissues can be broken down into three core groups: connective tissue proper, supporting connective tissue, and our liquid connective tissue. Now, with loose connective tissue proper, we're going to have two subcategories. One is loose, one is dense. The loose connective tissue and the dense connective tissue refers to how many fibers we have and the arrangement of those fibers to one another. With loose, we're going to have less fibers and have more ground substance or space between the fibers. And with dense, we're going to have more fibers and less space between the fibers. Supporting connective tissue, this is going to be our cartilage and our bone. And we will only look at compact bone today. We'll look at spongy bone and talk about spongy bone when we get to the skeletal system. And then we have our liquid connective tissue, which is blood and lymph. We're just going to look at blood because there's really nothing to look at with lymph, because lymph is essentially plasma. So if we look at it, it's just going to be clear because it's going to have the formed elements part, the erythrocytes, leukocytes, and thrombocytes. It doesn't have it. So we're just going to be looking at blood. So what that gives us is 11 different tissues to look at today. All right. 3 loose connective tissues, 3 dense connective tissues, 3 types of cartilage, compact bone, and blood. All right. So the first tissue we're going to look at is areolar connective tissue. And again, I'm going to be bouncing back and forth between the PowerPoint that I told you about on Canvas. I'll be bouncing back and forth between it and the lab manual here to talk about the different tissues and where we find them and what their primary functions are and how they're structured, and then go to look at the tissues so you can see the things that I'm talking about. Areolar connective tissue is one of the most widely distributed connective tissues in the body. When it's as widely distributed as it is, it also makes it one of the most abundant. When I say widely distributed, what that means is that it's found kind of all over the body. And that's the absolute truth, because areolar connective tissue is going to be found in the hypodermis. The hypodermis is the area that lies between the skin and the underlying tissue. So, when we think about the skin being attached to the muscle underneath it, that's going to be done using areolar connective tissue. Areolar connective tissue is going to primarily be made of collagen. The collagen is going to be arranged in a very loose pattern, but there are going to be plenty of collagen fibers, which makes this a very good tissue to use for binding purposes. Because we have collagen fibers running in so many different directions, and those collagen fibers are resisting tension, areolar connective tissue kind of works like the glue in our body. We see areolar connective tissue in basement membranes attaching the epithelial tissue to underlying other connective tissue, which is going to also allow for nutrients to diffuse through. The nutrients can diffuse through because of its loose arrangement of fibers. So when we talk about the integumentary system, and after this practical, we'll talk about how we're going to supply blood to the epidermis and the epithelium, and areolar connective tissue is going to play an important part of that process. So it has a lot of good uses, but again, most of it is going to surround binding tissues together. When we look at areolar connective tissue, we're going to see, like I said, these bundles of collagen fibers. Most of them don't stain very dark. The collagen fibers appear very, very light in the background, almost a pinkish color, sometimes a little bit darker, maybe a light purple color, but they're not going to appear as dark as the elastic fibers. The elastic fibers are almost going to look like pieces of hair that kind of got caught on the slide. They're going to be, in some cases, kind of, irregularly shaped. They're going to be black or kind of a dark purple, but it's pretty easy to tell the two different fiber types apart. We won't see the reticular fibers as that they don't stain on the areolar slide, so we're not going to see the reticular fibers on this particular slide. but we do have a small number of reticular fibers there. Fibroblasts are going to have an oval-shaped cell and an oval-shaped nucleus. There's one slide on here that you can see the fibroblast really well, and you can almost see the fibers that it's giving off the cell. Mast cells are a little bit harder to identify because they just kind of are round cells. So they just kind of look like a blob. And then we'll also see some macrophages, which are kind of a lima bean shape. Some of the slides have these identified. I don't think they're great, pictures of them. So, the last slide that we're going to look at really has a great example of a macrophage and a fibroblast. So that's the one that I'm going to.
Yeah, I was just about to ask that. It's kind of hard to see the differences between them.
Yeah, the fibroblasts, once I show them to you, they're a little bit easier to pick out than the macrophages and the mast cells. But I'll show you a picture of a mast cell where you can kind of see the bean, the lima bean kind of shape to them. So
Okay.
let's take a look at our first slide. All right, so this is our first areolar connective tissue slide. This is the PowerPoint that's on Canvas. You can see the majority of the fibers that we have here. They're kind of faded looking as they go in every direction. That's all collagen fibers that you see kind of in the background, kind of making this woven pattern that you see behind the majority of what is fibroblasts on here. You can see some elastic fibers, okay? These thin, dark, like almost hair-looking structures that we see woven across as well. Those would be elastic fibers. Now, right over here, okay, where this red arrow is, you can see this kind of elongated cell right there. That would be a fibroblast, okay? So you see another fibroblast right here where it's kind of elongated. We see another fibroblast right here where it's kind of oval-shaped and elongated. This would be a fibroblast right here. This green cell here, or the green arrow right here, this is a macrophage. This is not what I would consider a great picture of a macrophage because there's some other things that are kind of sitting on top of it. Again, so like on the practical, I like to put things that are clear. I don't want you to have to look at something and go, well, I don't know, that might be this. I guess it kind of looks like that. I want to pick something that at least is going to look, like it should for testing purposes. All right. And then the yellow arrow down here, this is pointing to a mast cell. Like I said, the mast cells are a little harder to pick out because they're just kind of a blob-shaped cell. There's not a whole lot of structure to them, but you can see that the nucleus is not quite as elongated, and we see the cell not being as elongated as we see with this fibroblast. In fact, with this fibroblast, you can see a little bit of that fiber coming right off of the tip of it right there. This slide, I'm not even that crazy about this slide, so I'm going to hop over this one because I think the next one is actually probably the best of the three. Again, with this, you can see all of the collagen fibers all running in different directions in the background. Again, you can see our thin, dark stained elastic fibers all cutting through the extracellular matrix. Here, this is a great fibroblast right here because you can almost see the fiber coming off of the fibroblast while you look at it right there. Here would be another fibroblast over here. Okay, this would be a fibroblast right there. There's another fibroblast right here. Now, this is the example of our macrophage. And if you look at the left side, you can kind of see how that left side is kind of concave and curved in, and how this side is concave and this side is convex. That's that bean kind of shape that we see with the macrophages themselves. Here's another one. This isn't quite as clear as the other macrophage, but again, you can kind of see that indentation on that one side. That would tell us that this is a macrophage. All right. Now, the next type of connective tissue is adipose. And you don't have to put adipose connective tissue. You can just put adipose tissue. You know, that some of these just, you know, when we talk about them in class, you know, we don't always say connective tissue after it. Some of them always we say connective tissue. You know, areolar connective tissue, elastic connective tissue, dense irregular connective tissue. But you don't have to say adipose connective tissue. You definitely don't have to say blood connective tissue. You know, you can refer to those, you know, as single names, and you won't, you know, you won't lose any points for that on the practical. Um, Adipose, again, is going to be our storage for triglycerides. Triglycerides are our body's long-term energy stores. You know, when we take in nutrients, there's three basic macronutrients, right? I mean, if you look at any label on any food that you take in, you see listed on there fats, carbohydrates, and proteins. Okay, and those are our three primary macronutrients. Our body uses these macronutrients for different things. Carbohydrates is our body's primary fuel source, or let me rephrase that, our body's favorite fuel source. That's why we have such carb cravings. But our body can't store carbs for long extended periods of time. So if we take in more carbs during the day than we burn during the day, then we convert what's left over to triglycerides. And those triglycerides are going to be stored in fat vacuoles, which we find in our fat cells, which are adipocytes. Adipocytes are unique, or not a unique type of cell, but they're a special kind of cell that we mentioned the first week, or was it last week? I can't keep track of all of them. When we talked about hypertrophy and hyperplasia, adipocytes are hypertrophy cells, meaning that the cells actually can get larger. The more triglycerides we store in the storage vacuole, the larger a particular adipocyte will get. As we burn fat, we will burn triglycerides out of an adipocyte, and the individual fat cells will then get smaller. But the triglycerides basically are going to push the nucleus and smash it over to the side of the cell. And the majority of what we're going to see is a hollow portion of the cell, which is the fat storage vacuole, where we would find triglycerides. Now, we can't see the triglycerides, you know, the light burns through them. So it just basically looks like a pink bubble. In fact, the adipose tissue, when you look at it, looks like a bunch of pink bubbles that have just kind of, been pushed together. We have two different types of adipose tissue in the body. When we're born, we have what we call brown adipose tissue. Newborn babies have this up until the point where they really start to develop more skeletal muscle. The more skeletal muscle they develop, the less brown adipose tissue we have. The body, it starts to atrophy in the body. And brown adipose tissue is a different color because it has increased amounts of mitochondria and a greater blood supply. The reason for that is because this tissue is actually used for fuel. Newborn babies lack the skeletal muscle to be able to generate muscular contraction to help them to regulate body temperature. So they have these fat stores that their body will actually use to create ATP, which is going to create heat energy given off during the Krebs cycle, which is going to allow them to help control their body temperature. This is the same thing that we see in hibernating animals when they aren't going to be moving around for long periods of time. Part of the reason why bears, bulk up before hibernation is not just because it's going to be cold outside, because they want the fat to insulate them, but they have high amounts of brown adipose tissue that's actually going to generate heat for them to help sustain their body temperature. Now, of course, as we grow and as we develop more skeletal muscle, we're not hibernating animals. So there's never a period of time when we're going to shut down and just rely on our fat to fire up and keep us warm. But we're going to rely more on skeletal muscle contraction. So that's why our brown adipose tissue levels start to diminish until they pretty much are gone. As adults, we're pretty much going to just be filled with what we classify as white adipose tissue. And white adipose tissue is going to be, like we said, our primary source for energy stores and triglycerides, but it's also going to serve as providing us a little bit of protection and padding, particularly around some of our internal organs. Now, is too much fat around the internal organs bad? Yes, it's not good, because then you're compromising the function of the organs themselves. But we need to have some fat around there because let's be honest, we get jostled around quite a bit, and those internal organs are shaking around inside of us, and we want to be able to protect those internal organs as they bang against the abdominal wall. The other place we're going to find white adipose tissue is in our subcutaneous layer of fat, which is also going to exist in the hypodermis. If any of you have done dissection, at some point in time where you maybe, did a rat dissection, or maybe you haven't ever done a dissection, but maybe you've cleaned animals, or I don't know, whatever. When you're taking the skin off the animal, there's a lot of white fibrous material that kind of is spider webby looking, which is the areolar connective tissue, which is what the glue that's making it really hard to get the skin off of the muscle underneath. But then there's also fat. There's also adipose tissue in there as well. And that's part of what gives our skin its puffiness, is this layer of subcutaneous fat, which we use to help regulate body temperature. The older we get, we tend to lose this subcutaneous layer of fat. And as we lose that subcutaneous layer of fat, we have a harder time regulating our body temperature, which is pretty self-evident if you've been around older people, and you looked at the thickness of their skin, and you also note what? They're always freezing cold, or they're always really hot. Okay. There's very rarely an in between. All right. My grandfather was, almost 90 years old when he died. And, for about the last year, you either went to his house and you either thought you were going to catch on fire, or you thought you were going to freeze to death. And most of the time, he thought both of those things within 5 minutes of each other. Because as they age and as we age, we lose the regulatory ability of maintaining body temperature, and that's one of the reasons why. It's not the only reason, but it's definitely a contributing factor to it. So again, there's lots of good reasons for adipose tissue. Again, just because something's good for you doesn't mean that excessively it's better for you. And this is a great example of that. But we do need proper amounts of adipose tissue. If your body starts to have too low a volume of adipocyte or of triglyceride storage, it goes into what we classify as starvation. Your body's going to burn carbohydrates first, then fats. And if we run out of fats, okay, your body will start to eat itself. And that's when we start to break down proteins. And that's always a bad thing. Okay. We don't ever want to use protein as fuel. Proteins are building blocks, not logs to throw on the fire. So what does our adipose tissue look like? Well, like I said, it looks like little pink bubbles. That's about as accurate a description as I can give you. And just about every slide you'll ever see of adipose tissue, that's what it's going to look like. Now, each one of these little bubbles is an adipocyte, all right? Here's the nucleus as it's smashed over to the side of the cell, and everything that you aren't seeing here, or you think you aren't seeing, everything here is the fat storage vacuole. And that is all filled with triglycerides, just like that one, just like that one, just like that one. So all the hollow that you see in here that doesn't look like there's anything there, that's all triglyceride storage. All right, and our last type of connective tissue, or loose connective tissue, is going to be reticular connective tissue. And what type of fiber do you expect us to see an increased number of with reticular connective tissue? It's not a trick question, y'all. We have
Reticular fibers.
There you go, reticular fibers, exactly. Everybody wanted to say it, but everybody was scared to say it. It's like, oh, that's too obvious. Yes, we're going to see more reticular fibers in reticular connective tissue. Now, like we said, reticular fibers are going to work, different than collagen fibers. They're going to be more structurally stable. We're going to use them to build things. And again, that's what we see with reticular connective tissue. We're going to find reticular connective tissue creating what we call the stroma of many organs. The stroma is the supporting tissue. It's not necessarily the tissue doing the job of the organ, but it's the tissue in the background that's giving the organ its particular structure. So we're going to find reticular connective tissue in things like the liver and the spleen and lymph nodes. Reticulocytes, which are going to be the cells that we associate with reticular connective tissue. Reticulocytes are actually going to be immature blood cells. They're immature erythrocytes, and eventually, as they mature, they will lose their nucleus and they will join the bloodstream as red blood cells. So when you look at reticular connective tissue, oftentimes students confuse reticular connective tissue with blood because they see all of these reticulocytes and they think they're red blood cells. No, they're not red blood cells yet. Okay, they will be eventually, but not yet. So how do you tell the difference between blood and reticular connective tissue? Well, blood isn't going to have reticular fibers, and the reticular fibers are these very thick, very branched black fibers. So if you see these branched black fibers, you know it's not blood, because you're not going to see any fibers with blood. So what does our reticular connective tissue look like? Well, to me, it looks like a cherry blossom tree. So if you've ever seen a cherry blossom tree, you may agree with me. All right. Cherry blossom trees have all these pink flowers all over them. And that's, to me, what this looks very similar to. You can see all these. What's that?
Right, quick question. I
Yeah.
thought lympho. Oh, I read in the notes that the main cell type is lymphocytes.
Or reticular connective tissue?
Yeah, it says it in the review notes. Yeah, it says the cells associated with the fibers are lymphocytes.
No, that's not right.
So I thought, yeah.
Reticulocytes.
Okay.
So I'll go double check. I'll go double check the review and stuff. That was probably a typo. So I'll check that out.
Okay.
You can see when you look here, you can see all of these reticular fibers. Okay, how they look like pieces of a tree. Okay, like branches or trunks of a tree. And then the reticulocytes that you see all scattered around them are going to be the flowers kind of the tree. So again, that's what it kind of looks like to me. And with connective tissues, you can kind of do that a little bit, where if it looks like something to you, jot it down in your notes and be like, oh yeah, that's what it looks like, because that may be something that helps you remember what it is. So if that type of learning works for you, then by all means, use that for the practical. This one over here, I don't think is as good. The reticulocytes didn't stain as well as this one on this side. I mean, we can see the reticular fibers really well, but the reticulocytes didn't stain as well over here. So this is definitely a better example of of reticular connective tissue. All right, so that takes care of our loose connective tissue. Next, we're going to move into our dense connective tissue. And when we talk about dense connective tissue, the first two we're going to talk about are going to be dense regular connective tissue and dense irregular connective tissue. Now, typically, I talk about these as a pair because regular and irregular are the descriptive terms that basically separate these two types of tissue. Both of these types of tissue are going to consist of large amounts of collagen fibers. So they're both going to be used to resist tension. But the arrangement of these collagen fibers is then going to dictate how we utilize these connective tissues. One of these is going to be very, very good at resisting tension in one direction, and one of these is going to be very, very good at resisting tension in lots of directions. Dense regular connective tissue is the one that's going to resist tension in a single direction. Because when we say regular, what we're referring to is the pattern of those fibers. They're all going to be running basically in the same direction. All right. So the collagen fibers in dense regular will all be moving in parallel bundles. We won't see any going this way and this way. Everything will just be running this way or this way or this way, but we won't see any crisscross of those fibers. Now, dense regular connective tissue, very strong in one direction, very weak in all other directions. Tendons and ligaments are what we're going to make or what we're going to make or use dense regular connective tissue to make. And tendons and ligaments are good at doing one thing, and that's pulling, okay, or preventing things from pulling apart. Tendons, we attach muscle to bone, okay? Muscles contract, they shorten, they pull on bone, they only pull in one direction. Ligaments attach bone to bone. They prevent that bone from pulling apart lengthwise in a single direction. Now, what these tissues are not good at is resisting tension in other directions. This is why we see ligamentous injuries occur when there's a shearing force that cuts across those ligaments, or a twisting force associated with those ligaments, because they're able to withstand force in this direction, but they're equally as weak in this direction. Dense regular connective tissue is going to look like this. So we have 3 slides here of dense regular connective tissue. 2 of them have fibers running parallel vertically, and 1 of them has fibers running parallel horizontally. Now, on all of these, you can see these little dark spots right through here. Those are the nuclei of our fibroblasts. On this one, it's a little harder to see because it's kind of washed out. This is not my favorite one. I like this one over here with C because you can really see the fibers in the directions that they're running, all parallel, and you can see these more darkly stained nuclei like this, right? But that's your dense regular connective tissue. Again, everything running in the same direction. You'll notice how much more tightly this is packed together than what we saw with, say, areolar connective tissue or with reticular connective tissue. All right, so it's definitely a much more dense pattern than we saw in those loose connective tissues. Now, with regular, we have fibers running all in the same direction. So with irregular, we're going to have fibers running in every direction. And this is what your dense irregular, that should say irregular, like it says there, dense irregular connective tissue looks like. All right, you can see those fibers. They're not all running the same direction. Some of them are running vertically, some of them are running horizontally. It just depends on where we slice through those particular fibers. Now, because this can resist tension in lots of different directions, it's going to be used in tissue that needs to be able to resist tension in lots of different directions. And so that's why we find this in the skin. The dermis of the skin, which is what the thickest part of our skin is going to be made out of dense, irregular connective tissue. That allows our skin to be pulled and stretched in lots of directions without tearing. Now, is this going to be strong in a single direction? Is it going to be stronger than dense regular? No, it's not going to be stronger than dense regular because it doesn't have as many fibers going in the same direction. But is it going to be more versatile than dense regular? Absolutely, because it can withstand all of the forces coming in lots of different directions. Now, this is going to be a highly vascular tissue. If we find it in the skin, we know that we're going to have the stratified squamous epithelium near it. So we're going to be providing nutrients to it. So when we get into the integumentary system, we're going to talk about dense irregular connective tissue again. And our last type of dense connective tissue is elastic connective tissue. So if reticular connective tissue is high in reticular fibers, we could expect that elastic connective tissue is high in elastic fibers. And that is 100% the case. When we look at both of these slides, you can see all of these darkly stained fibers weaving themselves through, all kind of in the same direction. Those are all of our elastic fibers. Now, those elastic fibers have the ability to stretch and become extensible and then be able to return to their normal shape. So we have certain tissues and we have certain organs in our body that we want to be able to have change shape and expand and then go back to their original shape. And probably one of the easiest things to think about is the lungs. The lungs, when we take a breath in, those lungs are going to have to be able to expand and enlarge and then be able to go back to their original shape and size when we blow out. So we're going to find elastic connective tissue woven into the lungs. We're also going to find elastic connective tissue associated with blood vessels, particularly some of the larger blood vessels like the aorta, because these blood vessels need to be able to stretch to accommodate blood flow and be able to snap back to their original shape. In the arteries, we're going to be able to change the size of the arteries in response to changes in blood pressure. So we're going to find elastic lamina woven into the blood vessels as well. So those are going to be the two primary places that we find elastic connective tissue. Now, don't just put elastic on the test, right? The reason is that we also have elastic cartilage on the test. So if you put just elastic connective tissue, I mean, you just put elastic and not put connective tissue, or you just put elastic and don't put cartilage, then I don't know which one it is you're talking about. So make sure you don't forget, if you write elastic, to at least give me connective tissue or cartilage afterward. Otherwise, elastic is going to get wrong. Not even partial credit, since I'm making a point to tell you about it. All right, any questions about the dense regular connective tissues? I know I kind of didn't hop back and forth between these just because everything I needed to tell you, you didn't need to see on here. Well, you can see this, the dense irregular. Sometimes people think it looks like steak, all right? So, you know, if you think that, you know, looking at the dense irregular connective tissue, if that looks like, you know, steak to you, then, you know, by all means, you know, use that as a descriptive term to help you identify the dense irregular connective tissue. All right, that takes us to our supporting connective tissues, and that takes us to cartilage. Now, we have three different types of cartilage. Each type is going to be used for a particular purpose. Each type is going to be, pretty easy to recognize, I think. And I think overall, cartilage is one of the easier tissues to recognize because cartilage is going to have some very unique features to it. And the main unique feature that it's going to have is its cell type and where those cells are going to reside. The chondrocytes and the pits that the chondrocytes live in, called the lacunae, are going to make cartilage pretty easy to identify. So that way, if you look at it and you say, hey, Dr. B said to look for this, now you've narrowed it down to one of three types of cartilage, and then all you have to do is look at the ground substance around it, all right? So I'll kind of explain that as we go through. Cartilage is a kind of like bone light, all right? That's kind of the way that I picture it in my head. We can shape things out of cartilage. We can use cartilage to build things. But cartilage isn't going to be as strong as bone. It's not going to be as supportive as bone. It's not going to be as protective as bone, but it's lighter and it's flexible. So, you know, typically when I'm thinking about cartilage, I think of cartilage as almost being like plastic, right? And I think of bone being more along the lines of like steel, okay? Meaning that bone is stronger, it's something that we can build, or we won't even go to steel, we'll say wood, all right? So bone is something stronger that we can use to really give us structural integrity, but it's going to lack some flexibility, and it's going to lack, and it's heavier, right? Now, with cartilage, we can build things that look like bone, but we're not going to be able to bear weight on those things because the cartilage just isn't going to be strong enough. But cartilage is going to be more flexible, and it's definitely lighter than bone. So that's why I always say cartilage is kind of like bone light. Now, hyaline cartilage is the most prevalent type of cartilage that we have, but it's also the weakest of the three cartilages. Hyaline cartilage is going to be found in a lot of different places. One of the primary places that it's found is on the ends of long bones, because hyaline cartilage kind of has an appearance and a feel to it that is kind of like plastic. Hyaline cartilage can be very, very smooth, particularly on the ends of long bones. And what that does is it gives us a nice, smooth, frictionless surface for those bones to be able to move back and forth on each other. We find hyaline cartilage in the trachea, creating the tracheal rings. So it gives the trachea some structure, but while making the trachea flexible so that as we move our head around, our neck can move. We find the hyaline cartilage at the ends of our ribs, connecting our ribs to our sternum. Why is that important? Well, the bones and the ribs, they're firm, they're hard. If we connected the ribs directly to the sternum, it wouldn't give us a whole lot of flexibility in the thoracic cavity. So instead, we have these costal cartilages, which are going to attach to the sternum so that when we take a deep breath in, the cartilage can flex and allow for our thoracic cavity to expand, allow for our lungs to expand. So, cartilage plays an important role in a lot of capacities, and hyaline cartilage is very, very versatile. Now, the cartilage itself, like we said, is going to be pretty avascular. The problem with it being avascular is that it is going to compromise its ability to heal itself. The nutrients that it is going to get is going to be from the perichondrium, which is going to surround the cartilage. This is a layer of dense, irregular connective tissue that's vascular, but it's also going to get its nutrients from the synovial fluid that we talked about in the synovial joints. So, the ability for it to undergo mitosis to be able to heal is going to be compromised by not having enough blood supply. Typically, when we damage cartilage, particularly in one of our joint capsules, the cartilage doesn't heal. Oftentimes, the cartilage will break off and it just kind of floats around in the joint capsule, and there's no going in and sewing the cartilage back together. It's going in and removing the cartilage oftentimes, because if we sew the cartilage back together, the only thing holding it together is going to be the sutures. It won't grow back together because it just doesn't have the capability of doing so. So when we look at cartilage, to me, cartilage looks like looking at googly eyes. The lacunae, where the chondrocytes are going to sit, oftentimes are kind of paired up next to each other, and with the nucleus in the middle of them, they kind of look like these weird shaped googly eyes. This is what your cartilage looks like. All right. So you see what I mean when I say googly eyes? Doesn't it kind of look like googly eyes? You got them kind of paired together. It looks like little crazy eyes. So if you look at these slides, if you look at any slide and you see these little googly eyes kind of staring back at you, then you know it's some type of cartilage, right? So now you've got a 1 in 3 shot of getting it right, because it's either hyaline cartilage, fibrocartilage, or elastic cartilage. And fibro and elastic cartilage are going to look significantly different to hyaline cartilage.
The perichondrium on the cartilage, that's the it's kind of hard to, I'm kind of having trouble seeing
Right
what
here.
that is.
Right here.
Where the a letter is? Okay.
Well, yeah, this whole thickness right here. Remember, the perichondrium is dense, irregular connective tissue. So in your mind, kind of picture what that dense, irregular connective tissue is that we just looked at, how it's going to have lots of collagen fibers. Now, particularly with hyaline cartilage, you can tell where the hyaline cartilage ends and the perichondrium begins, because hyaline cartilage in the extracellular matrix, look at how smooth the extracellular matrix is out here. We don't see hardly any fibers out here because there's going to be very few collagen fibers in that extracellular matrix. So all of this area around these cells where it's all smooth, this is all part of the cartilage. And then as we get out here and you can start to see the fibers, that's the perichondrium. Same thing over here. Same thing over here. Look where all this red is. Okay, that's just the staining for the extracellular matrix. But notice we don't see any fibers. But then when we get up here, you can see these fibers. That's the perichondrium.
Okay, thank you.
So that's how we're going to tell that this is hyaline cartilage, though. All right. Googly eyes tells us it's cartilage. Lack of fibers in the extracellular matrix tells me that it's hyaline cartilage, because your other two types are going to have fibers. And the first of those two types is elastic cartilage. Now, elastic cartilage is going to have elastic fibers. And we remember from the elastic connective tissue, those fibers typically stained dark black. When we saw the areolar connective tissue, those tissues stained dark black. Well, especially on this one over here, you can see all these dark black lines woven through this extracellular matrix. Those are the elastic fibers. We see our little lacunae with the chondrocytes sitting in them. Those are our googly eyes kind of staring back at us. That tells us it's cartilage. These black fibers tell me it's elastic cartilage. Now, elastic cartilage does have a perichondrium. We don't see it on this one. But if we come over here, we're going to see a perichondrium down here. Now, elastic cartilage, it has the ability to change its shape and return back to its normal shape. So where would we find elastic cartilage? What on your body has the ability to change shape no matter how you fold it and always pop back to its original shape?
The ear.
Yeah, your external ears, right? Go ahead, fold your ears up as many times and as many different ways as you want. No matter what your mama said about them staying that way, if you don't stop doing it, when you let go, they will always spring back to the original shape because of elastic cartilage. And our last type of cartilage, though, is fibrocartilage. Now, fibrocartilage, what do you think we're going to have an increased number of with fibrocartilage? Not a trick question. Fibers, right? Fibrocartilage means we're going to have more fibers than we've had with the other cartilages. So this is kind of like the tougher, stronger relative to hyaline cartilage. Where hyaline cartilage, our extracellular matrix was really, really smooth. With our extracellular matrix here, okay, here's our googly eyes. Our extracellular matrix here, we see all types of fibers. So we're going to have an increased density of collagen fibers going to be found in the extracellular matrix. Now, what this does is it makes it less flexible. So it's not going to be quite as pliable as the hyaline cartilage, but it makes it stronger and more wear resistant. You'll find as we go through the body, especially when we start talking about movement versus stability, and really that's the way anything is. The more stable something is, the less motion it has. The more motion something has, the less stable it becomes. So with this, we're creating more stability and wearability. So we're sacrificing some of that mobility. So we're not going to see bones that are connected by fibrocartilage have as much movement between them as bones that are not connected by fibrocartilage, okay, like our synovial joints. So where are we going to find fibrocartilage? We're going to find fibrocartilage in high load, high stress areas, like the spine. The intervertebral discs in the spine are going to be made out of fibrocartilage. The menisci in the knees, they're going to be made out of fibrocartilage. Why? The knee's under tremendous amount of stress and strain. We have hyaline cartilage in there on the ends of the bones, but we're also going to have a meniscus on either side to kind of cup and cradle and protect the ends of the femur as it smashed down into the tibia. We're also going to find it where the pubic symphysis is, which is where the pubic bones join together at the front of the pelvic girdle. Now, these are all high stress areas, okay? They're constantly in motion, they're constantly under load, and so we need to have something very wearable here because fibrocartilage has the probably one of the worst capacities for healing itself. And it does not have a perichondrium. All right, any questions about cartilage?
No.
All right. All right, so that takes us to compact bone. Now, like I said, we're not going to focus on spongy bone. We'll do that when we get to the skeletal system itself. But with compact bone, our location for compact bone is easy pretty much because it's found on the external surface of all of our bones. Every bone in your body, whether it be a long bone, short bone, flat bone, every bone is going to have compact bone on the surface. Now, when we talk about long bones, we're also going to find compact bone, though, in the shaft or the diaphysis of the bone as well. So that's going to help to aid in the strength of that particular bone. Because compact bone is really designed to do the two things that we associate with bone. One, it's designed to protect, and the other, it's designed to support. Now, compact bone is going to be a very vascular tissue, and it's going to have units of vascular units of functionality called osteons. Now, the osteons are going to have at the center of them a central canal. That central canal is going to be wrapped in rings of bone called lamellae. And within these lamellae are going to be osteocytes, which are going to be housed in tiny little pits called lacunae. So just like the lacunae we talked about with cartilaginous tissue, we're going to also find lacunae with compact bone. And that's where the osteocytes are going to reside. The lamellae, or these rings of bone, are going to have small little channels cut through them. These little channels are going to be communication channels from one osteocyte to another to allow them to share nutrients. And those little channels are going to be called canaliculi. So let's take a look at our compact bone, which to me, compact bone, again, is one of the easier things to identify because it looks like a tree, right? It looks like a tree stump or a tree branch that's been cut, that has the rings that you can see on the stump. Now, if we look over here on low power, we can see lots of central canals. All these little dark spots that we see through here are central canals. Those are running vertically through the compact bone. We also see right here what we call a perforating canal or a Volkmann's canal. That's running side to side between two central canals. But these central canals are going to carry things like blood vessels and nerve endings and lymphatic vessels. You can see the rings of bone wrapped around this central canal, and you can see from right here to right there, okay, this circular structure that wraps all the way around, that's multiple layers of thick, that is an osteon, right? So that whole thing right there is one osteon. From like right here to right there, that would be an osteon. From right here to right there, that would be an osteon, okay? So that's an osteon, the central canal along with the rings of bone around it. So if we come over here, again, we can see these rings of bone. Those are all the lamellae. Okay, our osteon would be from here all the way over to here with our central canal right in the middle. And you see all these little dark spots right here, okay? Those are the lacunae where the osteocytes are. And then these little, what look like cracks, those are the canals that run from one lacunae to another, right? That's what that little blue arrow is pointing to. Those are the canaliculi.
Okay, good thing, because that was kind of confusing. Before you went over it, yeah, when I was just reading it, it was just
right,
kind of confusing from the caniculi versus the lamellae.
The lamellae. Yeah, the
Oh,
lamellae
yeah.
are, like I said, they're the rings of bone. So each little ring you see wrapping around is a lamella. And when we get to the skeletal system, we'll do it again in depth. But the canals that connect right there, they look like little cracks. Those are the canaliculi.
Okay.
All right, and our last tissue type is our liquid connective tissue, and that's going to be blood. Again, blood's going to be made up of 4 different components. The greatest component of blood is actually the blood plasma. It makes up about 55% of the blood. And like I said, plasma is mostly going to be water-based. That allows us to dissolve substances in and transport substances easily throughout the body. The majority of our formed elements that we see on here are going to be our red blood cells, are erythrocytes. Okay, these are all of our erythrocytes. Okay, they look like these little red donuts. Notice they have a donut shape to them because they are lacking a nucleus. All right, red blood cells do not have a nucleus. We have these 3 cells here with these darkly stained nuclei. Okay, these are 3 examples of a leukocyte. All right, those are our white blood cells. And then this green arrow pointing to this little fleck right there, and there's a little fleck right there. There's a little fleck right there. There's a little fleck there, one there. Okay, those are our thrombocytes or our platelets. Now, the red blood cells, they're our primary oxygen carrier. Our white blood cells are involved in immune response, and our platelets, they're involved with clotting. Now, again, don't get this confused with reticular connective tissue. We don't see any reticular fibers here, okay? There's nothing woven in between these red blood cells. You're going to see some white blood cells. That should clue you in also that it's not reticular connective tissue and that it's blood. Any questions on our different types of tissue before I hop over to the repository? Because I'm going to show you some other examples.
You said red blood cell, that's the oxygen carrier. Leukocytes is immune response. What was the last one?
The thrombocytes, they help us help our blood to clot.
Okay, thank you.
All right, so let me go over to here to the tissue repository and kind of scroll through and grab a few examples of different connective tissues. Again, use this for the epithelial tissues as well. You know, I pointed out some really good examples of epithelial tissues last week. This is a good slide of elastic connective tissue. It's just not on high enough power. This one over here is a little bit better slide for elastic connective tissue. You can see all the elastic fibers woven through there, all running kind of parallel in the same direction. This is a good fibrocartilage slide. Now, I typically don't tell students to use the color of the slide when they're identifying the slide, because oftentimes you can get slides from different manufacturers that are stained differently. Almost every fibrocartilage slide I've ever seen has stained blue. So, if you want to use that bluish color with the pink stained nuclei, then or the chondrocytes, then you can use that to help you remember. This is a pretty good compact bone. All right, central canal here, central canal there. We've got lots of lacunae right there. If we zoom in on this more, we can see the lamellae wrapped around. You can see the canaliculi woven into and cutting through those lamellae. This is a good hyaline cartilage slide. We can see everything we talked about on this hyaline cartilage slide. All right, we have our chondrocytes sitting in these lacunae. Notice that there are no fibers in this extracellular matrix. Up here, that's our perichondrium. And then this is a good elastic cartilage. Notice all these black fibers, these dark fibers woven in with these chondrocytes right here. All right, that is definitely not hyaline cartilage. That is definitely not fibrocartilage. So it only leaves elastic cartilage. This is not the best areolar connective tissue. That's not great, dense, regular connective tissue. This is another good hyaline cartilage, just kind of like the one we looked at earlier in class. Again, this is your perichondrium over here. Not crazy about this adipose tissue. It's a little bit light. This is a pretty good areolar connective tissue. You can see lots of collagen fibers, lots of elastic fibers. I'm not sure. I don't think that's they have that labeled as a mast cell, but I'd say that's more of a fibroblast right there, given the long oval shape of that cell. Elastic cartilage, dense regular connective tissue. Here's a great slide for dense irregular connective tissue. And this actually puts into perspective too where we find dense irregular connective tissue. Because look, right here, this is stratified squamous that we see as part of the epidermis. So this is our skin right there. So that's the epidermis. That makes this the dermis underneath. And so you can see, again, the fibers, how they run in all different directions. So this is a really good slide of dense irregular connective tissue. Here's a reticular connective tissue slide. You can see again all of the reticulocytes and the reticular fibers woven through them. These aren't stained quite as bright as the other ones are, but that's a good slide to look at the reticular fibers. Fibrocartilage. So you can go through here and pick out some of the better ones that you see, like those. And those would be the types of pictures that you'll see, on the practical or something similar to those. Again, if it's something like this is really kind of too light. I don't like the way this is stained. Typically, we're not going to use something that's stained that faintly when we have a slide like this that we can use that's stained much more clearly. And this is all part of the skin and what we'll get into with the next chapter. All right. Any questions, y'all? I know I gave you a lot. Okay, I know we kind of hit the 11 tissues there pretty quickly, but I think, like I say, connective tissue to me is a little bit more easily recognizable than the epithelial tissue because it all looks different. All right. There's very few slides that look similar, so you can really focus on, all right, what are the characteristics? What do I see here to help you identify a particular tissue? I had a couple of people email me asking me, about flashcards, and yeah, I think absolutely. I think flashcards is a great way, or are a great way to study for the tissue slides, because you can put the answers on the back. You can look at the picture, print out multiple pictures of different types of tissue, shuffle them all up, and then just go through. And the ones you get right, stick in a pile. The ones you get wrong, stick in another pile, and continue going through them until you get them all right. All right. Once you get the identification right, then that's when you go back and master the function and the locations. Because again, the most important part is identify. Okay, because if you can't identify it, then it doesn't matter what function or location or cell or anything like that. It doesn't matter any of that if you don't know what the tissue is that you're looking at first. Any questions, y'all? All right, then. Well, that's all I've got. So again, next week, no class. You've just got Thursday to take care of your practicals. Following week, Mardi Gras. Enjoy your week. Okay, stay out of large crowds. I don't need anybody to get sick. And then I'll see you back here on the 25th, and we'll get into the integumentary system. I'll be in touch. I'll once I get your grades posted, I'll post an announcement, check on everybody next the following week during Mardi Gras. I'll have office hours on Thursday and Friday of Mardi Gras week, or Friday. I don't have any on Thursday, but I'll have some on that Friday. And if you need something, just reach out to me. If you need to check on anything, if you have questions about this stuff before Thursday next week, I've got a lot of office hours on Tuesday. I've got a little bit on Monday and Wednesday. So you can pop in during then and say, hey, Dr. B, I was studying this. I'm having some problems with this. Can you explain this again? And maybe I explain it in a way that, it clicks with you or something like that. So if you have issues, by all means, come by and chat about it. All right. All right, then.
All right.
Y'all have a good evening. Have a nice weekend. I'll study hard, and I'll talk to y'all soon.
All right. Have a good day.
All right.